In this section, we will be looking at the mechanisms of action of hormones. First, can you tell what is the intracellular signaling after hormone receptor activation? Well, almost without exception, a hormone affects its target tissues by first forming a hormone receptor complex. Formation of this complex alters the function of the receptor, and the activated receptor initiates the hormonal effects. Usually, there are three types of interactions that occur depending on the type of receptors present on the cell membrane of a target cell. These receptors are ion channel-linked receptors, G-protein-linked hormone receptors, enzyme-linked hormone receptors. We are going to discuss these receptors one by one. First, ion channel-linked receptors. Virtually all the neurotransmitter substances, such as acetylcholine and norepinephrine, combine with receptors in the postsynaptic membrane. This combination almost always causes a change in the structure of the receptor, usually opening or closing a channel for one or more ions. Some of these ion channels-linked receptors open or close channels for sodium ions, others for potassium ions, others for calcium ions, and so forth. The altered movement of these ions through the channels causes the subsequent effects on the postsynaptic cells, such as altering the cell's electrical activity, resulting in a response. Although a few hormones may exert some of their actions by activating ion channel receptors, most hormones that open or close ion channels indirectly do this by coupling with G-protein-linked or enzyme-linked receptors. So what are G-protein-linked hormone receptors? G-proteins are groups of cell membrane proteins called heterotrimeric GTP-binding proteins, and here the receptor where the hormone binds are known as G-protein-coupled receptors. These 7 transmembrane segments loop in and out of the cell membrane. The cytoplasmic tail of the receptor is coupled to G-proteins. G-proteins include trimeric parts alpha, beta, and gamma subunits. When the hormone binds to the extracellular part of the receptor, a conformational change occurs in the receptor that activates the G-proteins and induces intracellular signals that either open or close cell membrane ion channels, or change the activity of an enzyme in the cytoplasm of the cell. The trimeric G-proteins are named for their ability to bind guanosine nucleotides. So let's discuss how G-protein is involved in hormonal signaling. In their inactive state, the alpha, beta, and gamma subunits of G-proteins form a complex that binds guanosine diphosphate, GDP, on the alpha subunit. When the receptor is activated, it undergoes a conformational change that causes displacement of GDP by GTP guanosine triphosphate. Resulting activation causes the alpha subunit to dissociate from the trimeric complex and to associate with other intracellular signaling proteins. These proteins, in turn, alter the activity of ion channels or intracellular enzymes such as adenylylcyclase or phospholipase C, which alters cell function. No need to worry, these are second messenger mechanisms that will also be discussed shortly in this video. Now, if we need to terminate this signaling, it will follow back. So the signaling event is terminated when the hormone is removed and the alpha subunit inactivates itself by converting its bound GTP to GDP. Then the alpha subunit once again combines with the beta and gamma subunits to form an inactive, membrane-bound trimeric G-protein. Let me tell you another important point. Some hormones are coupled to inhibitory G-proteins, denoted GI proteins, whereas others are coupled to stimulatory G-proteins, denoted GS proteins. Thus, depending on the coupling of a hormone receptor to an inhibitory or stimulatory G-protein, a hormone can either increase or decrease the activity of intracellular enzymes. Moving forward, the third way of hormonal signaling is enzyme-linked hormone receptors. Some hormones use the enzyme-linked receptor tyrosine kinases for cell signaling. This is a cell where it is a transmembrane receptor, where the binding of an extracellular ligand causes enzymatic activity on the intracellular side. Activation of intracellular enzymes results in physiological effects. These enzyme-linked receptors are proteins that pass through the membrane. They have their hormone binding site on the outside of the cell membrane and their catalytic or enzyme binding site on the inside. The example of an enzyme-linked receptor is the leptin receptor. What is leptin? Recall. Leptin is a hormone secreted by fat cells and has many physiological effects, but especially important in regulating appetite and energy balance. The leptin receptor is a member of a large family of cytokine receptors that do not contain enzymatic activity, but signal through associated enzymes. This receptor exists as a homodimer. Homodimer means two identical parts. In the case of the leptin receptor, one of the signaling pathways occurs through a tyrosine kinase of the genus kinase JAK family, JAK2. We will focus here on the JAK-STAT signaling pathway. The binding of leptin to the extracellular part of the receptor alters its conformation, enabling phosphorylation and activation of the intracellular associated JAK2 molecules. The activated JAK2 molecules then phosphorylate other tyrosine residues within the leptin receptor JAK2 complex to mediate intracellular signaling. Gamma sites are specific tyrosine phosphorylation sites. Now comes STAT, which is a signal transducer and activator of transcription proteins. Well, this mechanism causes the phosphorylation of STAT3 proteins. After phosphorylation, it forms a dimer of activated STAT proteins. This goes to the nucleus of the cell, which then activates the transcription of target genes into mRNA to initiate new protein synthesis, resulting in physiological effects. So this was all about this hormone receptors interaction at the cell membrane, where we have discussed ion channel-linked receptors, G-protein-linked hormone receptors, and enzyme-linked hormone receptors. Now we will be looking at second messenger mechanisms for mediating intracellular hormonal functions. I hope these second messenger systems are very familiar to you. But now, we will discuss these second messenger systems for the main focus of hormonal signaling. When hormone bind to the receptor on the target cell, this is a direct effect of hormones on the receptor cell. Then, another way of hormones exerts intracellular actions is to stimulate the formation of the second messengers inside the cell membrane. We will discuss mainly three types of second messenger systems: adenylyl cyclase CAMP, second messenger system; cell membrane phospholipid, second messenger system; calcium calmodulin, second messenger system. So start with adenylyl cyclase CAMP, second messenger system. This is extracellular fluid and the lipid bilayer membrane of the target cell. Well, the binding of the hormones with the receptor allows the coupling of the receptor to a G protein. If the G protein stimulates the adenylyl cyclase CAMP system, it is called a GS protein, denoting a stimulatory G protein. Stimulation of adenylyl cyclase, a membrane-bound enzyme, by the GS protein then catalyzes the conversion of a small amount of cytoplasmic adenosine triphosphate, ATP, into cyclic AMP inside the cell. This then activates cyclic AMP-dependent protein kinase from an inactive state to active form, which phosphorylates specific proteins in the cell, triggering biochemical reactions such as the phosphorylation of proteins that ultimately lead to the cell's response to the hormone. Once CAMP is formed inside the cell, it usually activates a cascade of enzymes. That is, the first enzyme is activated, which activates a second enzyme, which activates a third, and so forth. This is known as cascading event of enzymes, which has its importance, as the slightest amount of hormone acting on the cell surface can initiate a powerful cascading activating force for the entire cell. But if the binding of the hormone to its receptors is coupled to an inhibitory G protein denoted as GI protein, adenylyl cyclase will be inhibited, reducing the formation of CAMP and ultimately leading to inhibitory action in the cell. Thus, depending on the coupling of the hormone receptor to an inhibitory or a stimulatory G protein, a hormone can either increase or decrease the concentration of cyclic AMP and phosphorylation of key proteins inside the cell. Now the second one is cell membrane phospholipid second messenger system. This is extracellular fluid in cell membrane. Some hormones activate transmembrane receptors of G protein that activate the enzyme phospholipase C attached to the inside projections of the receptors present inside the lipid bilayer of the cell membrane. This enzyme catalyzes the breakdown of some phospholipids in the cell membrane, especially phosphatidylinositol biphosphate, PIP2, into two different second messenger products, inositol triphosphate, IP3, and diacylglycerol, DAG. IP3 mobilizes calcium ions from mitochondria and the endoplasmic reticulum, and the calcium ions then have their second messenger effects, such as smooth muscle contraction and changes in cell secretion. DAG activates the enzyme protein kinase C, PKC, which then phosphorylates a large number of proteins, leading to the cell's response. The lipid portion of DAG is arachidonic acid, which is the precursor for prostaglandins and other local hormones that cause multiple effects in tissues throughout the body. Now moving forward toward calcium calmodulin's second messenger system. The second messenger system operates in response to the entry of calcium into the cells. Calcium entry may be initiated in two ways. One is the changes in membrane potential that open calcium channels. The other way is when a hormone interacts with membrane receptors, then it opens calcium channels. On entering a cell, calcium ions bind with the protein calmodulin. This protein has 4 calcium sites. When 3 or 4 of these sites are bound with calcium, the calmodulin changes its shape and initiates multiple effects inside the cell, including activation or inhibition of protein kinases. Activation of calmodulin-dependent protein kinases causes, via phosphorylation, activation or inhibition of proteins involved in the cell's response to the hormone. For example, one specific function of calmodulin is to activate myosin light chain kinase, which acts directly on the myosin of smooth muscle to cause smooth muscle contraction. It is an interesting fact that troponin C is similar to calmodulin in both function and protein structure. So this was all about the second messenger mechanisms, including adenylyl cyclase CAMP second messenger system, cell membrane phospholipid second messenger system, and calcium calmodulin second messenger system. Now move forward toward the hormone mechanism of action, which does not need to bind on plasma membrane cell surface receptors that shows hormonal action by intracellular hormone receptors and activation of genes. As we have also studied previously, steroid hormones and thyroid hormones are lipid-soluble hormones. So we can say these steroid and thyroid hormones are the hormones that act mainly on the genetic machinery of the cell. Steroid hormones act on cytoplasmic receptors and increase protein synthesis, and thyroid hormones act on nuclear receptors to increase gene transcription in the cell nucleus. After the hormone has entered the cell, therefore, it takes sometimes minutes, hours, or even days for newly formed proteins to appear in the cell and become the controllers of new or altered cellular functions. How do steroid hormones increase protein synthesis? Let's discuss this. A cell with extracellular fluid. This is the nucleus of a cell with a nuclear pore. Now the entry of steroid hormone is a lipophilic hormone. It diffuses across the cell membrane and enters the cytoplasm of the cell, where it binds with a specific cytoplasmic receptor protein. The combined receptor protein hormone complex then diffuses into or is transported into the nucleus. The activated hormone-receptor complex then binds with a specific regulatory or promoter sequence of the DNA called the hormone response element. As the combination binds at specific points on the DNA strands in the chromosomes and activates the transcription process of specific genes to form mRNA, the mRNA diffuses into the cytoplasm, where it promotes the translation process at the ribosomes to form new proteins. And how do thyroid hormones increase gene transcription in the cell nucleus? Well, thyroid hormones are also lipophilic hormones. But in this case, hormones first bind directly with receptor proteins in the nucleus. These receptors are activated transcription factors located within the chromosomal complex, and they control the function of the gene promoters, and in this manner either activate specific genes or the formation of mRNA. They activate the genetic mechanisms for the formation of many types of intracellular proteins, probably 100 or more. Many of these are enzymes that promote enhanced intracellular metabolic activity in virtually all cells of the body. Once bound to the intranuclear receptors, the thyroid hormones can continue expressing their control functions for days or weeks. So this was all about the mechanism of action of hormones, where we studied cell membrane hormone receptor interactions and second messenger mechanisms, and how steroid and thyroid hormones act mainly on the genetic machinery of the cell. Watch our medical videos anytime and anywhere. Download sqadia.com app now.